All right, thank you so much to Yossi and Julie and the committee for the invitation to be here. It's been a great learning experience for me hearing about all of the work from you all. So so I study cancer, but also neurodegeneration, because these are two of the diseases that are strongly impacted by DNA repair. And this is from a review that came out a couple of years ago about all of the hallmarks, molecular hallmarks of neurodegeneration. And most of the field studies these two related topics. So as we know, specific proteins aggregate or are dysfunctional in the most common neurodegeneration disorders in the human population. And the aggregation of those proteins, both inside the cell and outside the cell, have been postulated to really be the cause of the diseases. There is some controversy there, but there's excellent evidence from many different investigators that these effects, and also the effects of those aggregates on proteostasis generally, are the overwhelming agreed upon cause of neurodegeneration in humans. So it was gratifying, though, to see that nucleic acids have made it on the map now. And so DNA and RNA are also on this circle. And that's largely because there are many now, single gene disorders in DNA repair enzymes that cause neurodegeneration. And in many of those cases, neurodegeneration is not just one of the phenotypes, it's the phenotype of the disorder. So from that data, we know that DNA and RNA metabolism is very important for neurodegeneration. So my lab has studied the mechanisms of a couple of these disorders, which I'll just go through briefly. One general hypothesis we have, though, is that there are molecular connections between these DNA repair outcomes and protein aggregates and protein homeostasis. And so one of our theories is that these are not individual unrelated causes, but particularly that loss of DNA repair can in some cases lead to loss of protein homeostasis. So that's the hypothesis we're trying to test, and I'll give you two examples of disorders that I'll tell you about. One is AT, that Yossi has been so foundational in cloning the gene, and so many different papers from his lab showing the functions of this kinase. And then a second vignette about an enzyme called senataxin that is also responsible for brain-specific disorders. So as Yossi mentioned, so I work on the AT disease, ataxia-telangiectasia, which is a rare disease, one in about 40,000 live births, although about 1% frequency of heterozygotes in the population. It's essentially 100% penetrant. So if you have loss of function alleles of this gene, you will have this disorder, which has a lot of different aspects to the phenotype, but the primary one is a progressive cerebellar neurodegeneration that starts very early, age usually 2 to 3, and definitely diagnosis by age 5. And it progresses through early childhood, with the patients being almost certainly in wheelchairs by their teens. It also causes some other things, so increased cancer, very high lymphoid-related cancer rates, immunodeficiency, of course, radiation sensitivity, and premature aging. So ATM is one of the primary checkpoint kinases in human cells, so it's responsible for checkpoint activation following double-strand breaks specifically. And so amazingly, the children with AT who completely lack ATM also lack double-strand break checkpoints completely. And that turns out to be not essential for development. So it's really the other roles of ATM that come into play in terms of the brain disorder. Okay. So my lab has taken, oh, well, that looks kind of strange. My lab has taken a long time to look at how ATM is activated. The ATM is one of the master regulators of the DNA damage response. It has over 900 substrates. So some of these have been discussed at this meeting. P53, BRCA1, CHEK2, it's really the primary checkpoint kinase that is activating these tumor suppressors. So ATM is in the nucleus in most types of cells. It's a very large 350 kilodalton protein, which forms a 700 kilodalton homodimer. It is recruited and activated at sites of double-strand breaks by a repair complex that my lab works on called MRN. It's recruited there and activated through monomerization, and this promotes the stable binding of substrates by the ATM kinase, which then have many different functions in cells. Several years ago, we also found that ATM can be activated through a DNA damage independent mechanism that requires reactive oxygen species. This results in the covalent disulfide bond formation at the near the active site of the kinase that we now have some structural information on. This results in an active covalent dimer that phosphorylates many substrates and also can activate these pathways independent of DNA damage. And along the way in figuring these things out, we found a unique AT patient mutation, this truncation of the last 10 amino acids of the kinase. We've been collecting separation of function mutants to try to discern what these two different activation mechanisms do in cells. And it turns out that this patient mutation is a separation of function mutant, which specifically inactivates the ROS activation pathway. And these group of patients, there were 6 patients identified with this mutation, all have cerebellar neurodegeneration that is identical to the other canonical patients. So that suggests to us that the side of this pathway we really are interested in is the ROS side of the pathway when looking at the neurodegeneration part of the phenotype. So I won't show you any of this data, but we've used our separation of function mutants to really look at what ATM's functions are in cells depending on how it's activated. So if the ROS side of the pathway is inactive, but you still have DNA damage-related activation of ATM, we get cell cycle checkpoint activation through p53 and other tumor suppressors. We get end processing, which is a fundamental part of homologous recombination, DNA damage survival, and there's damage-induced autophagy that is also functional. If, on the other hand, we use separation of function mutants that block the DNA side, then we get a separate cell cycle checkpoint activation that's induced by ROS. ATM is involved in regulating mitophagy through still unknown functions. It regulates ROS in cells, primarily hydrogen peroxide, and we also see these protein homeostasis-related outcomes that we've been really interested in for the reasons I discussed at the beginning, because these are so crucial for neurodegeneration in the general population. So we've been focusing on protein homeostasis as an outcome of ATM loss for this reason. So I won't show you the data for all of this, but this is kind of our working model for how we think ATM deficient cells go from a normal state to what we think is a neurodegeneration prone state. So we noticed by accident that ATM deficient cells have this incredible widespread loss of protein integrity. So we see several hundred proteins aggregating in cells in culture that have lost the ATM protein. We know from experience in human neurons that we have grown in culture that the reason for this is increased ROS, so reactive oxygen species, mostly hydrogen peroxide, that result from ATM loss, are at the beginning of this pathway. That generates a transcription stress outcome that we know involves the accumulation of R loops. So these are 3-stranded structures where the RNA is stably base paired back to the template strand of the DNA, and the non-template strand is still present. We know that these R loops lead to single-strand breaks, not double-strand breaks, actually. And then following this, there is an activation of a group of enzymes called poly-ADP ribose polymerases, which you may know from cancer therapy. PARP inhibitors are used now in the clinic for homologous recombination deficient cancers. PARP is activated at single-strand breaks and also other DNA lesions. And we found that this hyperactivation of PARP is responsible for these protein aggregates that we find. So this is kind of our working model for how we get these aggregates, and just a little more detail on how PARP aggregates seed condensate. So there are others who have really established this field, but just to go over how this works. So in at sites of DNA damage, especially in levels when levels of high ROS are present, you get single-strand breaks and other form of damage. PARP enzymes, particularly PARP1 and PARP2, attach ADP ribose to other proteins as well as themselves at the DNA damage site. This modification can be made in polymers, so it's kind of a cloud of ADP ribose that actually resembles a nucleic acid in some ways. It has the same charge and some similarities to nucleic acids. We know from the work of other groups mainly that many disordered proteins are associating with these poly-ADP ribose polymers. And then we also know that ATM phosphorylation of many of these targets returns or removes these proteins from these ADP ribose clouds. What we think is going on in the absence of ATM is we have more damage because we have high levels of ROS. We know we have hyperparylation. That's been measured by us and by other groups at these damaged sites. The disordered proteins form these condensates, which are initially like liquid-liquid phase condensates at sites of parylation in cells. They're usually transient. But in the absence of ATM function, which normally removes these proteins from these sites, in the absence of ATM, these proteins accumulate. And we think that this is really the origin of the protein aggregates that we see in cells. And they're nuclear aggregates, they're not cytoplasmic. And we think they're associated with sites, physically with sites of damage. So at this point, when we were looking at this, we wanted to really validate whether this is real and whether this is relevant to the disease. And so we were able to acquire 21 autopsy brain samples from AT patients from the Neurobiobank. We got cerebellum samples and modified our aggregate purification procedure for these tissue samples. So basically a series of sonication and extraction and detergent extraction steps to isolate these aggregates and then quantify them by label-free mass spec. And so we did this with these human autopsy samples, and you can see examples here of the AT patients. We had 21 and 21 age-matched controls. There are really very few differences in the lysates from the cerebellum, but when we look at the aggregates quantitated here, there were over 1,000 statistically significant differences, and they're almost all to the right of this diagonal, showing that they're all in the patients. So we can see this happening in the patient brain. We also, for a subset of these, looked at the neocortex versus the cerebellum. So ATM patients have very few cognitive deficits. So the neocortex is almost untouched fairly normal in these patients, whereas all of the pathology is really located in the cerebellum. So we compared the cortex to the cerebellum, and the aggregates are definitely there at the site of pathology. So we know that they're there. The autopsy samples don't really give us causality, but definitely from the work that we've done in human neurons in culture, we think these are definitely there and likely acting at the sites of damage in the DNA. We've also looked quite a bit using that same tissue at bulk RNA-seq. This is an example with 5 AT patients and some controls. There's a lot of heterogeneity between the different patients here, as you can see, but there's this block of over 1,000 genes that is just dramatically reduced in the patients. And those genes, there are many genes in here, but what I've listed on this slide is there are 14 of those genes in this large block are independently associated with cerebellar ataxia in single gene disorders. So we think that this is, I think this is likely the proximal cause of the neurodegeneration is the transcriptional dysregulation that you get of all of these critical genes that are necessary for cerebellum function, or at least that's our interpretation of this. And if we look at the sites of transcription and where transcription is deregulated in the genome, I won't show you the data for this, but basically it's correlated very strongly with two things. The level of transcription in the wild-type cerebellum, so the patients have lost transcripts at the most highly expressed genes. At the low expression genes, the patients have often increased transcripts. So that's one correlation. And also the GC content in the promoter region is a very strong predictor. That's consistent with the idea that we're getting R loops, which are very sequence context dependent and occur at GC-rich sites. So our general hypothesis here is that loss of ATM creates this cycle of damage and transcription stress that occurs at specific sites in the genome where you have high transcription, high DNA damage, and these sites are the most important for the genes that are expressed in the cerebellum, and that leads to ultimately a degradation of function of those cells, and many cells are actually lost during the progressive neurodegeneration. Okay, so that was one quick vignette. The second vignette is about an RNA-DNA helicase called senataxin. We first started working on this because we're interested in R loops and how they play this causal role, we think, in the progression of events in AT. This is an RNA-DNA helicase, and basically we thought this would be the same story. So we're investigating this. So this gene, this is shown, the mutations in this gene cause actually two very different disorders. One is AOA2, which is quite similar to AT in that you have cerebellar atrophy in the brain, but other mutations in this gene cause ALS, where you have abnormal motor neuron function, and it's just not clear why different mutations cause the different disorders at this point. But we're interested more in the cerebellum side of this, and senataxin, as I said, is an RNA-DNA helicase. It's involved in transcription termination, RNA processing, as well as removal of DNA hybrids. It's conserved in lower organisms where it's been found to play important roles that are evolutionarily conserved in all these functions. So we wanted to know, basically, is senataxin deficiency or mutation similar to that of AT? And so we started looking at senataxin deficient cells. So this is a microirradiation experiment where we can see that you have more R loops at sites of DNA damage. This is a fluorescently labeled RNase H probe that we're using here. And then looking at R loops directly by, this is a blot for R loops as measured by an antibody that can be used to quantify R loops in cells. And we see there's definitely an increase that's reduced by overexpression of RNase H, which is an enzyme that's specific for hybrids. So we were kind of gratified to see that senataxin deficiency also causes problems with protein homeostasis. So this is work where we either deleted senataxin or truncated the N-terminal domain or the critical helicase domain. We're using a dye called proteostat here, which is an amyloid binding dye that allows you to look at amyloid formation in live cells and quantitate it. We're quantitating by FACS here. And so we can see with the depletion of senataxin, you get this increase in amyloid formation that's partial compared to the complete loss of protein homeostasis, which you get with a proteasome inhibitor. We also see the same things with N-terminal and helicase deletions, as well as with the mutations. So we've expressed full-length senataxin here, or full-length senataxin that is mutated with the mutations that are in the AOA2 patients. And you can also see that you see a higher proteostat signal with the patient mutations as well. We made a senataxin knockout line, and you can see that we get that same phenotype. So using our favorite aggregate preparation protocol, we looked at senataxin loss, and we were not surprised to see that in the absence of senataxin, you get very high levels of some aggregates. We're looking at TDP43 here and a truncation product. This is actually a gene that is involved in ALS, but we can see the aggregates very clearly in with the loss of senataxin. And you can see here also proteasome subunits are very high when we deplete the senataxin. This is actually in differentiated tumor-derived neuron-like cells. So when we look at the aggregates in these differentiated neurons in culture, we can see, or this is actually U2OS, the next panel is differentiated neurons in culture. In both cases, we can see that there are aggregates that come up primarily in the absence of syntaxin. There are several hundred of these, as you can show, as shown here in this quantitation. And we also used a live cell proteasome sensor. So one of the ideas with loss of, or the accumulation of aggregates in cells is that what this causes is an overload of the proteasome capacity. So we used a proteasome live cell sensor to look at this, where this is showing the sensor and its output related to a control fluorescent protein. And we can see with the loss of syntaxin, we get this increase in this ratio, which means there's a lower proteasome capacity that we also see in the knockout. And then we also see a decrease in growth rate, which is consistent with this loss of capacity as well. And then we see an unfolded protein response. So this is a response to having too many unfolded proteins in cells, and there's a well-characterized response that we're using the CHOP protein promoter as a reporter here driving M. cherry. And you can see that this is hugely increased when we lose the syntaxin protein. So there is this correlation between R loops and aggregates. We know that this is direct because when we show these aggregates in cells, we can get rid of the aggregates by overexpression of RNase H. So this is an alternate way of getting rid of R loops in cells, and we can get rid of the aggregates with RNase H, which means that the R loops are driving the aggregate formation. But when we started looking at the mechanism of this, we tried some things that we learned from our ATM experience, and we found that it didn't really work. So PARP inhibitors, which eliminate ATM-related aggregates, had no effect here, and also giving cells the antioxidant, which completely blocks aggregate formation with AT, just totally didn't work. So we knew that we had something distinct from what was seen in ATM deficiency, so we had to sort of think about what that might be. And after a while, we realized, maybe we should think about what is in the nucleus more broadly. In fact, that if you look at where syntaxin is in the cell, it's actually in the nucleolus. This was already known from yeast and from mammalian cells, but thinking about this, we realized, well, maybe we should think about the nucleolus. That's where the enzyme primarily is. And we started thinking also about this work. You may not be familiar with the work of Stephen Lee, but he's shown that in the ribosomal DNA loci, so this is a depiction of the ribosomal DNA. This section is the intergenic spacer, where it's not where the ribosomal RNA is actually produced. And he's shown in his series of papers that the ribosomal intergenic spacer is a place that gives rise to these non-coding RNAs that are highly repetitive and are stress-specific. And so if you give heat, you get some RNAs. You give acid, you give a different RNA. And what we found was that syntaxin loss actually gives rise to a specific subset of these non-coding ribosomal RNAs from the intergenic spacer. Stephen's work has also shown that what these RNAs can do under some circumstances is seed aggregates and phase condensates of specific proteins. And so ultimately what we went on to show is that the RNAs created or generated from the ribosomal RNA, specifically the intergenic spacer, are recruiting these proteins, and they become kind of stuck there in the absence of syntaxin function in a manner that's dependent on the R loops that are formed. And we think that these are the condensates, not general nuclear condensates, but nucleolar condensates that are really recruiting the protein aggregates that we find in this scenario. So this is kind of our working model at the moment. So similar to ATM and yet quite different, specific to the ribosomal DNA, but still involving this nucleic acid that's produced, and accumulation of proteins to this nucleic acid is what we've identified as really the key event in this case. So with that, I think I'm on time, which is good. So I'd just like to thank Philip and Nicolette, who are really key in doing the final parts of the ATM work that I've shown you, in addition to these critical former members, and Shumei Wen was really the driver of the Xenotaxin work, and I'd be happy to take any questions.